Thank you to the Moving the Needle corporate sponsors, Home Infusion Options and Metro Infusion Center, and our exhibitors, Takeda, Janssen, and Bristol-Myers Squibb. You share our mission of advancing patient care and make events like this and our grant making possible.
Our next speaker, unfortunately, could not make it because of a conflict. His name is Jun Huang. He is an assistant professor in the Pritzker Molecular Engineering program. And I think we have a video, is that correct, of his presentation. So let's get that to roll.
Hello everyone, this is Jun Huang from Pritzker School of Molecular Engineering. My laboratory makes molecules and cells for treatment. Currently, we are developing CAR T cells as a treatment for inflammatory bowel disease. I want to begin with a brief introduction of inflammatory bowel disease, or IBD, which is a chronic disease characterized by inflammation in the digestive tract. There are two types of IBD. Ulcerative colitis and Crohn's disease. The symptoms are uncomfortable and can be serious. Unfortunately, there is no cure for IBD yet, but there are roughly 7 million patients worldwide who suffer from this disease. Previous research has shown that IBD patients typically have too many Th17 cells. These are a type of inflammatory cells that are useful at normal levels, but can cause IBD at high levels. IBD patients also tend to have very few Treg cells, which are the cells that prevent overinflammation. To regulate levels of Th17 cells, we drew inspiration from CAR T cell therapy, which has been successfully used to treat certain blood cancers. In this process, a patient's T cells are extracted from their own blood. The T cells are then functionalized with a protein called a CAR, designed to target and kill cancer cells, then are injected back to the body as a cancer drug. We aim to use a similar process to treat IBD. We strove to design a CAR T cell to target and kill the Th17 cells in IBD patients. Th17 cells express a protein called IL-17A, so a CAR T cell that recognizes this protein would be able to hunt Th17 cells to restore a healthy level of Th17 cells in IBD patients. Recently, we successfully engineered a CAR T cells that recognizes IL-17A. We also showed that our CAR T cells do not induce off-target inflammation, which is a promising result. The next step in our research is to show that our CAR T cells specifically recognize and kill Th17 cells. In conclusion, with CAR T cell therapy, we hope to restore a healthy balance between Th17 cells and Treg cells in IBD patients to treat the painful symptoms. Finally, I would like to thank all the audience for listening. I would also like to thank the GI Research Foundation for supporting our research. Finally, I wanted to thank Annie Mitchell and Nick Aspey for working on this project and the entire Huang Lab for the support. Thank you.
So just to say a word about this is a type of genetic engineering. Basically, you're taking a patient's cells, genetically modifying them, and then putting those cells back into the same patient so you don't get a rejection. And you're instructing that cell to home into the site of inflammation where it secretes an anti-inflammatory molecule that is very important in rebalancing the immune system. So it's really a very cool technology, and we're very fortunate in having Dr. Huang work on it.